All right, guys, how's it going? Welcome to EOC Review 3. This is kind of a big day here. We got photosynthesis, cell respiration, DNA, mitosis, and protein synthesis. A lot of stuff going on here. You guys should have your EOC review sheet out in front of you. Please follow along with me as I talk about this. And you guys can highlight stuff. If you want to grab a highlighter, you can hit pause and go grab one. You can write notes off to the side. But it kind of might help you, again, follow along what's going on. As I said before, these videos aren't that exciting because you are just staring at a screen, and I know that can be very tricky. Trust me, I struggle with it. But it is basically what I can do for you. Okay? All right. So I hope these reviews are going well for you too. All right. So let's boogie. Let's do this. All right. So we're going to talk about photosynthesis first and foremost. You guys know I love me some photosynthesis. All right. The whole point of photosynthesis is to make glucose. Glucose is better known as the food. Please make sure that you know this formula, C6H12O6. It is a carbohydrate. Okay. How does this happen? Basically, plants need to have water and sunlight and carbon dioxide, and all those things come together to make food. I love this picture, as weird as it looks, but basically it is summarizing photosynthesis in a whole. Okay? All right, so organisms that undergo photosynthesis, we got your plants, okay? Plants can involve all sorts of different things. You got grass, you can have this is a plant, this is a plant, and this is a plant. This is a tree and a moss. And if you guys remember, I think I told you guys that I had a kid argue with me once that a tree was not actually a plant. And it is, all right? So they're going to be undergoing photosynthesis, okay? Bacteria are going to be undergoing it as well. If you notice, everything I'm showing you here is green, okay? And that's kind of a good indicator there. All right, and we're going to have some protists, for example, green algae. We call them phytoplankton that actually undergo photosynthesis. Photo means light, okay? All right, so where is this taking place? We're going to do a whole zoom thing here. We're going to start right here. Here's your producer or a plant, okay? The bulk of photosynthesis, hopefully you guys remember, is going to take place inside of a leaf, okay? And actually what I'm doing here is I'm getting a cross section of a leaf. So basically I'm cutting it and flipping it up, okay? And this is what it looks like, okay? And the bulk of the photosynthesis is actually going to be occurring right in these cells right up here, okay? My avid kids, you guys learned what those were specifically. EOC is not going to ask you that. All right, just know that it's in the upper part of the leaf. Okay, where does it occur in the upper part of the leaf? It's going to occur in a chloroplast. All right, I bet money they're going to ask you that one. All right, chloroplasts basically are going to have this cool little structure right here. Inside of it, it's going to have a pigment known as chlorophyll. Now, if you noticed, or if you remember, I said in looking at all those organisms that undergo photosynthesis, how they're all green. That's because of chlorophyll, which is the main pigment that's inside of there. Now, what exactly is a pigment? A pigment is present in plants, and what it does is that it absorbs that sunlight energy. Classic example, how many of you guys have ever worn a black shirt on a really sunny day and you go outside and you get really, really, really hot? Well, basically, that black shirt is absorbing every wavelength of light. It is absorbing that sunlight energy, which therefore causes you to warm up. We can't do anything with that, but plants can take in that sunlight energy and actually make glucose from that. Okay. Now, the summative formula for photosynthesis, and actually before I show you guys this, you guys learned photosynthesis way more complicated than what the EOC is going to ask you. You learned about the Calvin and the light-dependent reaction. For photosynthesis here, basically all you need to know is the equation, okay, and chlorophyll and the chloroplast, that kind of stuff that I just talked about. All right, so again, like I told you guys, we got to have carbon dioxide, we got to have water. And what we're going to make is we're going to make glucose and we're going to make oxygen. Okay, sunlight energy can also be placed right up here. Think of this as a reactant. Okay, basically what is needed. So imagine that plant stirring that pot of food, right? We had sunlight energy, water, and carbon dioxide. Products are what they're going to make. If I were to make an EOC for a bunch of high school kids, I would say, which of the following is a reactant for photosynthesis? And I would list all sorts of stuff. Like I would list carbon dioxide, glucose, oxygen, carbon dioxide, and maybe some other random thing. So know this formula, okay? And that's all you need to know for photosynthesis. Pretty easy. All right. So let's talk about cell respiration. All right. Cell respiration is kind of the opposite of photosynthesis. Think of photosynthesis as making the food. Okay. Whenever you make the food, we got to break it down. Okay. So the whole point of cell respiration is to break down glucose into that usable form of energy, which is known as ATP. Okay. Something that we got to break it down into this form so that we can use it. Okay. Our body's pretty complicated. All right. Now, who's going to be, let me back up. What is ATP? ATP is a nucleic acid. Okay. Nucleic acids, again, are going to have CHON and P. And I don't think that you really need to know this, but definitely know that ATP has phosphate in it. Okay. Does ATP have energy? And the answer is yes. Okay. It is loaded up. And a lot of it has to do with the molecular structure of these phosphates. We're not going to get into that because I know the EOC is not going to ask you that. I'm just going to try to cover what I think the EOC might ask you, but just know that ATP is associated with energy. Now, here's the cell respiration, the summative formula for this one. If you guys notice, it is the exact opposite of photosynthesis, where in photosynthesis, these guys right here were the products. They made glucose and oxygen. We need these and to fuel this whole process. While what we're going to make is CO2, H2O, and ATP, which is basically a form of energy. Okay, these are the reactants in the products. Again, know this formula. Definitely know the relationship between oxygen and carbon dioxide for both of these. In photosynthesis, plants take in CO2, release oxygen. In cell respiration, we take in oxygen and basically exhale carbon dioxide. Now, what's cool about this carbon dioxide, guys, is where is it coming from? It's actually coming from the food that we eat. So anytime you're exhaling, you're breathing right now, you're exhaling that Bojangles biscuit that you had this morning, or you're exhaling the delicious quesadilla that you had in the lunchroom. I don't know, okay? But you're exhaling carbons from the food that you consumed, which I think is really freaking awesome, okay? All right, so we have two different types of respiration. We have something known as anaerobic respiration. This is respiration that occurs without oxygen. So oxygen is not needed to fuel it, okay? Anaerobic. Organisms that are going to be undergoing this is basically every single living thing in the whole entire world, which is really pretty cool, okay? So from a bacteria cell to us, to a plant, okay? Plants are going to be doing this as well, okay? So how does anaerobic respiration work? Basically, we got our simple organisms right here, and I don't know why I have the complex over there. It's all good. So they're going to take in the glucose. They're going to eat the glucose. They're going to break it down into pyruvate, okay? And they're going to generate some ATP. This whole step you don't need to know, okay? Key thing that you need to know about anaerobic respiration is that when simple organisms undergo that, they make a little bit of energy, but they basically, this whole business that they produce right here, the pyruvate, is they don't need it anymore. This ATP that they make is enough energy for them to live, okay? So this is given off as waste. So fermentation is basically where the remainder of the sugar of glucose is basically given off as waste, okay? It's fermentation. Now, we have two types of fermentation. Hopefully I didn't talk about it up here, and I didn't, okay? The two types of fermentation are going to be lactic acid fermentation and alcohol fermentation, okay? And simple organisms undergo that. We do not undergo, well, we can go undergo fermentation, but it's not ideal, okay? All right, simple organisms, I don't care about that. All right, so how do we work with anaerobic respiration? We eat, this zebra here is eating food. You guys eat a Bojangles biscuit. It has sugar inside of it. We break it down into this pyruvate. Again, you don't need to know that for the EOC. We generate some ATP, but this one ATP is not enough energy for us, okay? We have to generate a whole lot more. And these are all your complex organisms. And what I mean by complex organisms, I mean anything but bacteria or yeast. It's everything else, okay? All right, so what they're going to do is they're going to send this pyruvate to the mitochondria. And if you guys recall from the video from yesterday, is that mitochondria is basically where respiration takes place. And this means aerobic respiration. It's where we make ATP. It's the powerhouse, okay? All right, aerobic respiration occurs with oxygen present. You got to have oxygen in order to fuel it. If you don't have oxygen, you can't do that, okay? All right, only organisms that undergo aerobic respiration is all the complex organisms, which basically means everything but bacteria and yeast. So this includes all the animals. It's going to include plants. It's going to include protists. It's going to include fungus, okay? Basically everything that you see in this picture is going to be undergoing aerobic respiration, okay? Where does it take place? In the mitochondria, like I said before. This was a slide that you saw in the video from yesterday, okay? So how does it work? Basically, if you recall, aerobic needs oxygen. So this pyruvate is what we made in sorry, brain fart here, in anaerobic respiration. Think of it as our sugar broken down into a different form, okay? Oxygen is basically the key to the mitochondria. If we don't have this oxygen, this structure right here will not be able to get in there. But once we have that oxygen, we're going to make all this ATP, okay? The other reason why bacteria and yeast don't undergo aerobic respiration is because they don't have a mitochondria, okay? So that kind of eliminates that process right there. Now, the key thing here, what they will ask you is they might ask you what generates more ATP. Is it going to be anaerobic respiration or aerobic respiration? And the key thing here is that it will be aerobic respiration. That's going to generate the most ATP, okay? All right, that is cell respiration. And again, you guys can hit pause at any point in time, take a stretch break. I know I'm talking pretty fast, but hopefully you're hanging with me, okay? All right, switch gears. Let's talk about DNA. All right, DNA is deoxyribonucleic acid. They're not going to ask you that, but it is a nucleic acid, and the monomers are called nucleotides. Okay, now nucleotide, it looks like this. Okay, and it's made up of several things. First and foremost, this little guy up here is a phosphate. This part right here is going to be our sugar, which is deoxyribose, and this part right here is going to be a nitrogen base. Okay, we can have 4 basic nitrogen bases: adenine, thymine, cytosine, and guanine, A, T, C, and G. Okay, so this is going to be the structure of a nucleotide that makes up DNA. Okay, all right, so with these nitrogen bases, they pair in certain ways. Okay, A will always base pair with T, and G will always base pair with C. Okay, kind of an important thing to remember here. Okay. All right, so the structure of DNA, if we were to break this baby down, if you notice, I have 6 nucleotides making this baby up. There's my nucleotide, my phosphate, my sugar, and my base, okay? So there's your phosphate. Nitrogen bases make up the steps, which hopefully I show in this one, but I don't think that I do, and that is deoxyribose, okay? So the steps of the ladder are going to be your nitrogen bases, while the handles that you hold on to are going to be deoxyribose and phosphate that are alternating. So phosphate, sugar, phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, base, base, base. Notice the A and the T is paired together, okay? So if I were to give you this one right here, okay, what would be the corresponding matching bases? If A, let me hop back to this one right here. Remember, A likes T, G likes C, okay? So right here, we would have a T, right here, we'd have an A, and continue on down, okay? So very important thing to remember, all right? So this is DNA, all right? Okay, so what is a gene? A gene is basically a part of DNA that's going to code for a trait, okay? It's found within our DNA, and it's basically spelled out by our nitrogen bases, okay? Which I have right there. The nitrogen bases are going to spell out the directions, all right? So we're going to hop to protein synthesis right now, okay? So in protein synthesis, we're going to have something called RNA. RNA is basically the same thing as DNA, except it's just a tad bit different. We have some differences, okay? An RNA nucleotide, basically going to be the same thing where we're going to have the phosphate. We're going to have a different sugar. This one's going to be known as ribose. I don't think the EOC is going to ask you about the sugars, okay? But they do care about the phosphate and they care about these bases. The other difference is that in RNA, instead of having T, we're going to have uracil. Think of them as basically like twins, okay? Where they both are going to be base pairing with adenine. So T likes adenine and uracil likes adenine, okay? It's just that U is found in RNA while the T is found in DNA. I noticed in looking at some of the work that you guys were giving me that you're getting these all mixed up, okay? And it's not that difficult, it's just a matter of paying attention, okay? Paying attention to detail, okay? All right, so how is DNA different from RNA? The double helix has 2 strands, and RNA is simply going to be 1 single strand, okay? DNA has deoxyribose while RNA has ribose. And probably the biggest one is that DNA has thymine, adenine base pairs with thymine, and RNA adenine base pairs with uracil. They'd be stupid if they didn't ask you this, okay, on the EOC, because that's a pretty big one. It's a really big one, okay? All right, so we have 3 types of RNA. All right, the first one is called messenger RNA. Messenger RNA is basically going to be copying a message from a gene on DNA, okay? Like this lady, this was the DNA. What's going to shoot out there is the mRNA. It's a copy of it, okay? The next one is going to be tRNA. tRNA is the carrier or the transferer, I guess you can say. It's going to be carrying amino acids to messenger RNA. Don't ask about the picture, okay? If you notice I left out rRNA, that's because the EOC is not going to ask you about that, okay? All right, so let's talk a little bit about protein synthesis. All right, what we're going to do here, and what you guys can see is here's my cell, there's my genes inside of the nucleus, the chromosomes. Basically, from the DNA, we're going to make proteins from that. Okay, that's the whole point of protein synthesis. All right, so protein's going to be made from a gene on DNA. Okay. All right, so brief summary of protein synthesis. The very first step is going to be transcription. The second step is translation. Transcription, translation. These get a little bit confusing sometimes, okay, because they sound so much alike. It's just a matter of learning them. And also, if you put them in alphabetical order, transcription always comes first, okay? So if you notice, I'm going from DNA to RNA there. What we're doing in transcription is that we're making messenger RNA, plain and simple, okay? All right, so the transcription animation, I do actually want to show you this one. Nice. How did I know that was going to happen? Give me a second here. Okay. If we were to watch this one, okay, and they're not going to ask you step by step of this one. They're mainly just going to ask you about the end result. But we're going to have an enzyme come and attach itself to the DNA. This is a gene, okay? And what it does is that as it moves down, it's making that messenger RNA. You don't need to know the name of the enzyme for the test. You did for me. Okay, so this is the messenger RNA that's being spit out. Now, guys, pay attention to the U's, okay? If you notice here, CG, CGAU. Anytime we deal messenger RNA, this is when you're going to have the U's involved, okay? And that's it. You got your messenger RNA made. Done, okay? All right, that's ugly. Okay, where's my PowerPoint? All right, so that's transcription. So if we were to transcribe the following, okay, and again, I'm only showing you one strand of DNA. We only copy one strand of DNA because mRNA is single-stranded. So again, T will be A is going to be U, and C is going to be G, and continue on down. All right. How you know not to use T's in this one? Because I'm asking you to transcribe, and hopefully you can associate that with protein synthesis. But also, the only time you're going to use T's is when I go DNA to DNA. When you're going from DNA to RNA, that's when the U shows up. Okay. All right, so next step is translation. We made the messenger RNA, now we got to do something with that. Where does translation take place? It's going to be in the ribosomes. If you guys recall from yesterday, ribosomes are going to be the location of protein synthesis, okay? So what we're doing here is we're making a protein, okay, for messenger RNA. That is what's happening in translation. So DNA, transcription, we got our RNA, translation, protein, okay? All right, so I'm just going to click through this one really quickly, okay? Here's the messenger RNA that we made earlier. You're going to have a ribosome basically attached, but before I do that, you have codons and anticodons. Codons are going to be found on messenger RNA. It's a lot of memorization, you guys, okay? And I'm trying to give you hints along the way to help out. All right, so ribosome's going to come and attach, pow pow, okay? And tRNA is going to fly in, okay? Bloop. All right, tRNA looks really crazy, okay? It's this weird little spaceship looking thing. So this weird thing that it has up here, if you guys recall what I just said, is that tRNA is going to be bringing the amino acids to DNA. This is your amino acid right here, okay? This right here is known as an anticodon, okay? Basically, there are 3 base sequences on the mRNA and also on the tRNA. The anticodon is on the tRNA, okay? Very simple way to match those up. If you guys notice, RNA to RNA, you're going to have the U's involved, okay? The only time you're going to have T's is in DNA, all right? So it matches up. So got another space, another one's going to fly in, match up, okay? Notice how it does that. So peptide bonds are going to form between these, and the first tRNA is going to leave, and the ribosome is going to shift, okay? And it's basically going to continue on, all right? I'm not quite sure how the EOC is going to ask you about this, okay? What they can do is they might be most interested in how you got the protein, okay? So, for example, they could say, hey, here's a strand of DNA. What amino acid sequence does it code for? Okay, and this is what I noticed you guys screwing up on your tests. Well, I have this DNA. I got to go to mRNA first. Okay, so again, when I'm doing that, I have the A equals U and the G equals C. So we're going to get this one right here, okay? And that's just finding the complementary bases. I tell kids oftentimes when you do your EOC, you're going to get a scrap piece of paper. What I would do is I'd write down little hints, and probably one of them that I would write down would be DNA, and then say A equals T, G equals C, and then go RNA and say A equals U, G equals C. Just to have that as a reference. I would also write down those summative equations for photosynthesis and cell respiration. And just have those as a reference, okay? But I digress. So I still haven't made a protein. So what you got right here is you got this doodad right there, and then you can give me the amino acid sequence. Now you guys have, and let me find this for you guys right up here. You guys got this sheet right here. why did I do amino acid sequence? I meant to do chart. Sorry, I don't have this up. Okay. You guys got a very much bigger chart than that one that I'm showing you right there. See, look, EOC review, it's on that one. Okay, you guys got this chart right here, all right, which shows you basically how to find those amino acids. And they'll provide this for you. You don't need to know how to do that. But for example, if I have AUG right here, how you do this is my first base is A, okay? You can put your little finger on that one. Second base is U, and it'll be in here. This is the met that we got, okay? If you guys don't like doing the first base, second base, third base, just look for the sequence in there, okay? So for example, my next one is CCU. I know it's not as efficient, but if you look in here, where is CCU? Sometimes when I'm feeling lazy, I do that. You guys have probably seen me go over it 12 times. CCU, whoop, proline, okay? And that's all you got to do. So in order to use this, this is for the messenger RNA codons. I got to do this very first step here, getting into that messenger RNA, and then you can move forward with that. Okay? All right, very important to know that. They will ask you to do that for sure. Okay? All right, let's move on. Let's talk about mutations. Okay? Mutations is basically going to be a change in the DNA sequence. Okay? It's where it's just not going to be the same. We're going to have a little mistake in there. Now, what can cause that? We got two things that can cause that. We can have damage to the DNA by UV radiation, which is sunlight, through smoking, through chemicals, all sorts of crazy things like that can actually damage that DNA, okay? Also, it could be just mistakes in DNA replication, okay? Very simple thing, okay? All right, so we have two basic categories of mutations. We can have a point mutation. This is going to be a change in one base. So for example, if you notice, I kept these in groups of threes because back in protein synthesis, we have codons and anticodons, all that good stuff like that. So the fat cat ate the wee rat. If I were to change one letter in the sentence, let's say I change that C to an H, the sentence still kind of makes sense, but not really. The fat hat ate the wee rat. So if you were to read that, you'd kind of be like, what? Okay. Point mutations can be really bad, all right, but they're not the end-all be-all. An example of a point mutation is actual sickle cell anemia. This is where, and you guys learned about this yesterday as well, or the video before, hemoglobin is a protein found in our red blood cells that's all bound up like this. If you have sickle cell anemia, basically what happens is that that protein is not nice and round, it's flat, okay, which causes the red blood cells to basically stretch out. And that is due to a point mutation. Okay, so right here, that is the point. You have one different amino acid, which causes the hemoglobin to be all jacked up. Okay, all right, frameshift mutation is the addition or deletion of DNA, which results in a completely different sequence. Okay, these can be a little bit more deadly. So go back to our sentence, the fat cat ate the wee rat. Let's say that I deleted that C. Okay, I took the C out. If I take that C out, I can't just have at and then ate the wee rat. Everything has to stay in groups of threes when we talk about DNA. So if I were to delete that C, this A is going to shift over. Okay, and the T is going to shift over, and the W is going to go over, the R is going to go over, and you're going to end up having these little stragglers here at the end. So we have the fat, added T, U, at. It doesn't make any sense. Okay, that's why frame shifts can be pretty nasty. The example of a frameshift that we talked about was Tay-Sachs. Tay-Sachs is terrible, terrible. If you notice, this is the normal amino acid sequence here. It's completely different, okay? Tay-Sachs was basically where the brain starts to swell and getting overloaded with fat because you're not producing the enzyme to break down that fat in the brain, which can be pretty bad, okay? All right. Shifting gears, done with mutations. Let's talk about cell cycle summary. Let's review the cell cycle, okay? All right, so the cell cycle, or mitosis, is going to be taking place in our body cells. Now, where are we? Here's this goofy dude, okay? There's the tissue. Remember, we saw this in our last video, and there's our cells. So all the cells that make up the body of an organism is better known as a body cell, okay? All right, so what is the total number of chromosomes in our body? We're actually going to have 46, okay? What is the whole point of the cell cycle? The whole point of it is to grow, to replace old cells, or it can actually be a means of reproduction for simpler organisms, okay? Like this guy right here. This is a hydra, the same group as jellyfish and sea anemones. This right here is the baby shooting off from it, okay? It's asexual reproduction. All right, so where does the cell cycle take place? It's going to take place within the body cells of an organism, okay? The first step of this is something called interphase, okay? Interphase, if you guys see this on your little packet there, highlight it. Did I put that in the packet? I'm sure I did. If I didn't, somebody should punch me. I didn't. You guys might want to write this down, okay? Everybody find this sheet right here. You might want to write down interphase, blah, blah, blah. There's not a whole lot of room, but you need to jot this down somewhere, okay? Because this is huge. You'll see this when you start answering questions here in a second, okay? So the first step of the cell cycle is interphase. This is cell preparation, okay? Now, interphase is actually made up of 3 different stages. We have the G1s and the G2. This is just growth. That's why we have the G there. The one that the EOC loves for some very strange reason is S. So on your EOC review, you can put like number 1, interphase, cell preparation, summarize this, and write down the S stage. This is where DNA is replicated, okay? You're going to see that they're going to ask you a lot of questions on that. I don't know why, but they will, okay? So DNA is copied. The whole point of the cell cycle, again, guys, is to basically make more cells. I can't just take the cell and chop it in half. It doesn't make a whole lot of sense. If I were to chop it in half, I might have half DNA over here, the other half over there. I might have some mitochondria there, some over there. Whatever's in this cell right here, I got to have exactly the same thing in the new cell. Okay, and I should have said that before I started to talk about interphase. So that is why we're doing this cell preparation and replicating the DNA. Okay? All right, so let's talk about mitosis is number 2. Mitosis is basically where the replicated DNA is going to be separated. Okay? And is basically made up of 4 stages. Now, these 4 stages, you don't have to write these down because the EOC is not going to ask you about that, which I think is ridiculous. What they might do is they might give you the pictures and you might have to put them in order, okay? Which I don't know how you can do that without knowing the names. But mitosis is where the replicated DNA is separated. Now, if you guys notice, this is how interphase looks, okay? Interphase is the longest phase, this is what our cells look like the majority of the time. And this looks different than that one, okay? So if you notice, the DNA is actually winding up. That's what's happening in prophase. The next thing that happens, they're going to line up in the middle, okay? They're pulled apart, my favorite. And then we got basically where they're going to divide, all right? I think even if you didn't know anything about this, you guys could probably put these in order, okay? So again, it goes squiggles, line up in the middle, pulled apart, and then looks like they're actually divided, okay? That is mitosis. Now the final stage is called cytokinesis. This is where everything that we made in interphase is going to be split off into these new cells. Everything is going to divide, okay? And the end result, 2 identical cells. Identical is the huge, 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 huge thing. There is no difference, okay? There is no difference. So if an organism has 50 chromosomes and it undergoes mitosis, how many be present in the new cells? If it's identical, it's going to be 50. It's going to be exactly the same, okay? If it had 100, how many would it have? It still would have 100, okay? So easy enough, okay? So that was a whole lot of talking. What you guys are going to work on is you're going to work on these review questions. Hopefully you guys are putting some effort and some time into it and thinking about these things. Like I said before, I had kids in the past that failed basically all 3 terms with me and decided, hey, I don't want to take biology again. And so they busted their tail on these reviews and did all the work. Not only did they do the multiple choice, but they actually did all these things as well. And they killed the EOC in a good way, and they passed, okay? So if you guys have struggled a couple terms, don't write this off. There still is hope, okay? And to be honest, the EOC is typically easier than the tests that I give you, okay? All right, so take some time to answer these questions. Once you're done with these questions, hop on up, and I actually want you guys to start working through these, okay? This is a good review as well, all right? We'll go over these answers for EOC Review 4. Okay? Hope all is well, and I'll talk to you guys later. I'll see you for EOC Review.